Excellent. Well, first, huge thanks once again to Johns Hopkins University for sponsoring this. Big thanks to Joanna and to Daria for facilitating everything, being the brains and mechanics behind it all. And welcome back if you're returnees to this workshop series. This is number 4 of 6. And I'm super excited because fresh from Cuba, and then by way of Minneapolis, of all places, is my dear friend Damon Kylo of 31 years and 2 months. I first met him on a tropical island where we were both conducting scientific research, Barrow, Colorado Island in Panama. And Damon made his way all the way out here to the Driftless Zone to join us to talk about broader outreach. So I'll have Damon introduce himself briefly, and then I'll dive into examples from each of us.
All right. Thanks very much, Barrett. We've been collaborating for a long time. So this is just one more thing that we're doing together, but a new thing for us to do this type of thing together. Yes, my life has taken me from the U.S. to tropical climates as a PhD researcher. So I was studying mutualisms for some time with the Smithsonian Tropical Research Institute. And at some point, life and coincidences took me on a path away from that academic research life to educational outreach and using illustration and my scientific tools to collaborate with other scientists in order to tell their stories. And I'll talk to you after Barrett talks about his stuff, so I'll see you soon.
But first, we've got a fishing bat stenciled shirt by Damon Kylo, as well as dun, 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 Cerambycid longhorn beetle shirt, also by Damon Kylo. And we've got an octopus shirt by Damon Kylo.
A favorite of mine, yes.
Love it. Many of you know me through the previous workshops. I'm a professor of entomology and animal behavior at the University of Wisconsin-La Crosse in the driftless zone of Wisconsin. And I'm interested in the intersection of science and art. I'm interested in cultural entomology, and my scientific research ventures into sleep and social insects primarily. Now, what I'd like to do is go through 4 examples of how I've worked on visualization with others, and then Damon will share his personal examples of working with others to reach broad audiences through scientific visualization. And then we'll have a participatory exercise. So step 1, my friend Christian Rabeling discovered a new ant, which isn't a big deal because there are tens of thousands of species of ants. New ants are being described all the time, but this one was not only a new genus, but a new subfamily that was sister to all the other ants. So it solved some mysteries about the evolution of family Formicidae. And he said, Barrett, can you draw this ant for PNAS? And I thought, okay, geared toward peer-reviewed published science. And ultimately, to maybe my surprise, but not Christian's surprise, this ant became famous because it solved some mysteries about ant evolution. And so E.O. Wilson published an image in his Letters to a Young Scientist, and it's been featured elsewhere. So what started out as a figure for an exclusive group of scientists ended up being used and perpetuated through popular science. So how did I approach this? Christian and I had 24 hours together, and he told me about this ant. He said it has no eyes, it has no frontal lobes. He thinks because it's pale and blind that it's probably fossorial, burrowing. And I sketched out some ideas. He wanted to picture it as it might be seen in its natural habitat. And I came up with this drawing initially, which he okayed after a back and forth. And then I thought it'd be fun to take photographs of textures of soil and impose those within using a raster-based program like Photoshop or GIMP. And I added a couple more layers, used a vector-based program just to get the scale bar and the lettering. And this ultimately was the image published in Wilson's book. So here we've got an image that no one has really seen, but might give an idea of how this ant survives. Okay, so that's case study number 1. Case study number 2. John Abbott, an amazing dragonfly damselfly researcher now in Alabama, overlapped with me at the University of Texas, Austin when I was working on my PhD. And probably unwisely, I decided along with my research, I was going to illustrate his Damselflies of Texas field guide. Now, this required a very different approach than an illustration using the microscope, a one-off with Martialis eureka, the ant from Mars for Christian Roebling. In this case, I needed to do hundreds of illustrations of 76 species and multiple versions of each. So this is more like an assembly line. And because John Abbott is appropriately very persnickety, he would say something like, hey, that damselfly looks great, but the average one is a little bit more yellow, or maybe more pruinosity, waxier. And thanks to working digitally, I was able to make changes by the power of layers. So John invited me to write a few pages about the process. And largely, I was inspired by Ed Lamb, who's done exquisite books like Damselflies of the Northeast and a new field guide on dragonflies of North America. And I combined approaches of Ed Lamb, Steve Buchanan, and a few touches of my own. So I described that process in these few pages, and I'm going to go through a stepwise progression to give you an idea of what our pipeline was like. Here's a dead damselfly, really dead, faded, brownish. The eyes are dead. It's crumpled over. The legs are curled. It just looks dead. How do you add vivacity to this? Well, what we decided to do was disassemble into component parts the wings, the legs, the head held by wax at 2 different angles, and then the thorax with the abdomen or separately. And those scans, as you can see, the backgrounds are different shades of gray. It's a problem. So what I need to do is clean up each of those component parts so that when I reassembled them, the contrast, brightness, everything matched. When I assembled them, I had to build parts that were missing, like spines on the tibiae, maybe fill in veins, clean up areas. So there's a lot of digital cleaning. And there are a lot of layers here. Right now, there may be dozens of layers in this image of a male damselfly in dorsal view and lateral view. Now, watch carefully because it's going to brighten up with lots of colors. And I'm pulling these from photographs of live specimens. So bing. So you've got lots of bright colors in the swamp damsel, but it's lacking a little bit of 3-dimensionality. It's a tubular organism. So if I add a little streak of diffuse white and a little streak of darkness on the underside, and I'll do this with several other forms of this species, now you can begin to see some shape, some 3-dimensionality to that. The final spread for this particular species would look like this. So even though it's one species, you know you have males and females, you have juveniles, sometimes you have different forms. So this one page on the left of this spread should guide you to the variation that's available in nature of the red-tipped swamp damsel. So that was the field guide example. Now I want to flip it and talk about how I hired others. I wasn't working for a client, but here I was the client. And for the Insect Epiphany, my book that just came out, I was working with a number of people, but most directly with my mother and the artist who produced this beautiful cover and several full pages inside, Elizabeth Jean Younts. So there were back and forth there, lots of stories there to produce this and other images. But I decided instead to talk about working with my mom, Karen Ann Klein, on a book in progress. And it's tentatively titled Sleep Across the Animal Kingdom. We have no idea what the cover will be like or anything else, except that The Harvard University Press editor is reading it right now, but I'll share an example of one of the images she produced. Now, starting, I said, Ma, I'd love to. By the way, this is a collaborative book with amazing sleep researchers Niels Rattenborg and John Lesku. And we thought it would be exciting to have a tree of sleep, basically a phylogenetic or evolutionary tree of relatedness that shows animals and highlights those for whom we've studied sleep, which means also highlighting those lineages of animals where nobody has studied sleep. So this is really complicated. Can't give an artist that to work with. So I neatened it up a little bit. And then I removed all those names and focused on the individuals that I'd like Karen Ann Klein to feature. And note that you've got this branch from sponges, cnidarians, up to vertebrates and invertebrates. Invertebrates largely empty, and that's a big part of the story. Well, my mom produced this beautiful drawing, the beginning draft, and I okayed it. I okayed step by step, and then this full drawing. And then she started to detail it with the owl. And so here we're working on birds who sleep. And I made corrections and touched up areas where I wanted additional empty branches to show other lineages. And here was the final question mark product. Gorgeous, right? The problem is in the early drawing, I didn't catch that there wasn't the big split as in my phylogenetic tree of vertebrates and invertebrates. So this was a major change, unfortunately, that my mom had to correct. Now, she doesn't work primarily digitally, so she went and erased and reworked and came up with this beauty. Now, this could have been the final with some of my digital touch-ups, except for the fact that we had to add an amphibian right there in the center. So she drew it separately, and I digitally imposed that frog. So here's a back and forth between a scientist and an artist who happen to be son and mother. I thought it'd be fun to also show an example that's very different. I used to work for a natural history display making studio and then the American Museum of Natural History making exhibits. And those included three-dimensional models like this HIV and others. And it included life-size insects, giant marbled orb weaver, spider consuming me, etc. So with each comes a different challenge. And I'm going to share one work that isn't my best, but it poses challenges that lend for good discussion between back and forth between scientist and me. And I did this for Chase Studio, and Chase Studio was working for the Museum of the Rockies. And one diorama demanded that we produce extinct fish. And this was my task. I was given this white sheet with a fish species that hadn't even been named yet, an extinct shark, an Iniopterygian, only nicknamed Iniop1. So I have some details about it, or thoughts about what it might have been like when this was alive. And then this poor photograph of a single fossil, obviously of a single sex of this fossil fish. A wonderful illustrator, David Calamine, produced this field sketch style illustration of the fish in lateral view, as he presumed it might have looked like. So imagine this challenge. You take this lateral view of someone's sketch, this dorsal view of a fossil, a photo of a fossil, and now you have to produce a three-dimensional model. So I used those and scaled my wax sculpture in pieces. So first I did a lot of research to figure out, okay, what was this fish really like? Who are the closest extinct relatives? Who are the closest extant or presently living relatives? How can I go piecemeal to produce the body, the fins, all the accoutrement of this Iniop1? And note that I'm trying to match this illustration when I'm producing my prototype. Well, Richard Lund, who subsequently described this species in the scientific literature, visited us upon invitation to fact check or be a quality control. And he went one after the other to each of us to talk about our models. And he looked at mine and he said, nope. Basically, what you see here with those tubercles shooting up, he suggested, no, they probably went down. In fact, he was quite sure they went down. Maybe it was like a catfish bottom dweller, and it would sift the benthos to get food. So now I had to rework. I tore the prototype apart a bit and reworked it, and then molded, casted the items, the elements, and then put those cast components together with a fake set of eyes, and Brian, a colleague, painted it. So I wondered 30 years later, how is this held up? Does it still exist? So I did a quick Google search, and sure enough, at the bottom back of this diorama, there she is, at least since 2017. So one example of, again, a back and forth between a scientist and an artist collaborating requires this research at both ends. And I'm trying to get a hold of Richard Lund to find out if indeed this model still holds up scientifically. So those are my examples.
All right. Well, let me introduce myself a little bit more before I get into my examples, because I have arrived to a place that I don't think too many people arrive to as I did. My path was very much an academic researcher. So for the first half of my life, I'm living in the US, I'm going to graduate school. I get excited, as many people would, from living in a temperate zone, that the tropics holds wonders and amazing diversity. So I decide I want to go to graduate school to learn about tropical ecology and become a tropical ecologist with the opportunity to go live and work in a tropical environment. So I do that. I spend many years at the Smithsonian Tropical Research Institute. I'm studying mutualisms. Particularly, I'm studying mycorrhizal fungi and how these fungi exist and within a plant, and they work together in order for the plant to exist and for the fungus. And I'm fascinated by mutualisms in general. But at some point, life and coincidences is taking me away from that academic world and making my life more transient. I'm moving to different places, and I start to do educational outreach. So now, instead of being the scientist who's researching, I am the scientist who's talking with other researchers and helping them share their stories to a broader audience. Their attempt to do educational outreach. Most scientists don't want to do their own educational outreach because it's not their forte and it's not their interest. Just the opposite for me. I love telling the story and I want to help with that process. So the images you see here are just a wide variety of the ways in which I am going about through the past couple decades now of doing educational outreach. You know, from things like you might expect, a figure illustration within a journal, but also using my contacts with scientists to actually see what a leafcutter nest or a fungus farming ant nest looks like on the inside. So I have an idea of really what's going on there, so I can make this image that's on the top center. And had the chance to go to Bayreuth, Germany, of all places, for a garden festival in order to do interpretive signs for the university in the area to highlight their research for this setting. And below, some illustrated books, which I'll go show you a few examples of. And finally, on the far right is murals. I've been doing a lot of murals. That's a way to tell a story about the natural world to a very big audience who just happens to pass by. So they're definitely not a niche audience. They're whoever passes by. So I don't know if it's clear from my explanation, but the process for me is that science informs my art. I need to know a lot about what a scientist wants. So when they contact me with a project, the first thing I need to do is read their manuscripts, read their research about that particular project they want me to work on, or be able to look at what they want me to show. And in turn, the art reveals the science. This slide shows a graph down on the lower portion. That's the synthesis of the researcher for what they see as a hypothetical model of how the leaf cutters exist in their world with their fungus and with their plants. So they've reduced everything down to this graph, but in order to help the general audience understand that better, we need to blow it back up out to the real world. And I needed to illustrate all the elements of what goes into being a fungus farming ant in terms of harvesting leaves, in terms of choosing which particular species of plants to use. And they don't do that on their own. They do that with the help of the fungus. They're in communication with their own fungi that they're growing. And the fungus is telling them through chemical signaling, nope, I don't like that one, or yes, I do like that one, go get some more. So it's a very complicated world. And that graph is showing all of that in some method, but unless you are a niche scientist for that, you wouldn't understand it. So he needed me to elaborate the whole picture for the broad audience. So in order to reach a broad audience, I want to clearly state right now, reaching a broad audience for Barrett and I is not to tell you about social media and such methods to expand that message out. It is to explain science in a method that is more understandable for people who aren't particularly working on a particular area. A scientist might work on one tiny little part of this world, and their colleagues are very interested in that, but there is a broader audience that also wants to understand it. So I use various methods. Again, I've talked about those, and we'll go through some. So scientific figures, the first one. I better make this slide a little more interesting if I want to be who I am. So this is the way I want to present this slide. That scientific figures, yes, they should be very clear and concise. This is a life cycle that's showing a Metarhizium pathogenic fungus that's infecting this poor little grasshopper, or this lucky grasshopper to help Metarhizium exist. And I like this particularly of many of the illustrations I've done because it just turned out to be artistically beautiful. It tells a story clearly and concisely, yes, but it also is visually impressive so that the reader wants to take a closer look and learn more about it. Many times you can't work in color. Color is a very powerful tool, but a lot of figure illustrations based on cost especially need to be done in grayscale or black and white. I'm not a scientific illustrator. I wasn't trained to be a scientific illustrator in terms of using all the techniques that are used in order to use just black and white in order to show a beautiful image. So in this image, this figure, the author could have shown just those two graphics with the call, the echolocation signal of the bat. Yes, and that would be fine for an audience that is only researchers who have studied echolocation in detail, know exactly what those different axes mean, and what the call might be expected to look like and how they vary. All right, but no, this is for a general audience. This author wanted to share with the people something about the whole system. So we have two different types of bats. We have an aerial hawker and a substrate gleaner. The aerial hawker, you can see in the silhouette next to the name, has very thin wings. Those wings help it to move gliding through open air spaces very quickly and rapidly, gulping up insects along the way. And it could be lots of insects within an hour. Substrate gleaners, on the other hand, they're trying to pick and choose an item that they want to consume, but they have to go through all that vegetation that's within a tropical rainforest. If you haven't ever been to a tropical rainforest, it is completely different from a temperate forest. In a tropical rainforest, there are plants growing at all levels from the ground up to the canopy, and there's almost impossible to move through that ecosystem if you don't have a machete. And of course, the bat has to use its wide wings to be able to migrate around pursue its prey and capture the prey using echolocation. Now the reader has a full understanding of the facial physiology to show how that varies for the type of echolocation they're using, the wing morphology, and they have the call right there together. So they can see all of it together to understand the whole story. If you're lucky, you reach a broader audience simply by the serendipity of an editor of a journal saying, hey, we'd like to have your particular article represented on the cover of our journal. That's not so simple in so many ways. It might have to do with the colors that they want in the journal. No, that didn't work for us because last month's journal looks like this and yours won't contrast enough. So in any case, there's a lot of luck involved. But if you do get the chance to create a cover art, then you have the chance to reach a broader audience simply because they're going to see that art and not just go directly to the article that they want to read. All right, so now I'm going to show you a particular example for a figure, step by step, and talk about the process that I had to go through with the researcher. As I said, I'm a scientist. In this case, Rachel Page, a bat scientist, presented this to me. She said, here is a map, a phylogeny of a bunch of bats. Okay, tell me more. Well, I want to tell something about their diet choices, what they choose to eat, and how that's related to how they're related to other species. If you look at this very busy map, it's pretty hard to see anything. You can barely read the text, and it's just a busy item. Scientists do present these in talks, and you wonder how they expect you to read it. So the first step was just to say to her, I need to do something about how that appears. One, we need to be able to read the names and have more space between those names so that someone can possibly actually see what's going on. And I would prefer to put them in a straight line fashion. So, okay, that we have a very clear view so that you can quickly look through it, find the bat you're interested in, and look at it, see how it relates to other bats and what they're eating. The next step for me was to say to her, listen, I don't know anything about bats. Not really. I'm not a bat researcher. Where does this little group of bats that you're showing me fit into the big scale of bats? Can we add something more for a map of where we are in the world of bats for the general audience? So on the left side, you see these are the big groups of different types of bats. The figure's only going to focus on one of those five, the Phyllostomidea, which she would say are the cute ones because they have big ears and a big leaf nose in order to have this very advanced echolocation system. And then on the right side, you see this highlighted area in black. Those are the species that she said, these are the ones we're focusing on. There's a lot more of these Phyllostomid bats, but just this group we're going to be looking at. So then you have the finished figure that was printed in the journal on the left. And you can now see that there's some color there. Okay, what is the color doing for us? It is bringing your attention to that. Yes, there's a lot of information to see there. Now we have the map of where you are in the world of bats. You see the highlighted area is enlarged, so it's looking at that specific part of the Phylostomids. And now what is this colored area? Oh, right, this is telling me what the bats are eating. So they're eating insects or amphibians or reptiles, birds, mammals, or plants. In this case, some fig fruits. And they're color-coded so you can quickly look across the species. So you might look at two different bats. If you look at the top group, if you look back on the left on the map, you see that they're very differently separated from the other group. They're actually part of a totally different branch. So you wouldn't expect them necessarily to do the same things. In any case, the nice thing for me as a collaborator in this was that after Rachel Page was able to look at this, she said, wow, because you've made this so clean and clear and I could see it all together in one spot, I became aware of different things that I wasn't aware of in the past. It has opened my eyes to a little bit more of my own bat group. Nothing better could possibly happen in terms of a collaboration. Except maybe this. Because me as the artist, I want to do something exciting as well as nuts and bolts of making science clear. I want to create some beautiful art as well. And she said, okay, now we have that image that's telling us about the diet choice. I want to show to the readers the full range of size of the bats in that group they were looking at. And that is a major difference, going all the way down from the Micronycteris on the upper left. It's less than 10 grams, all the way up to this giant Vampirum bat on the right, which you can't see the full body. It's enormous. And then showing them what their diet choices are in a size relationship. So if you're a tiny bat, do you only eat tiny things? Well, it turns out no. If you're that tiny Micronycteris, you might be eating that large reptile that it has in its jaws. And you can see that from the figure how it expands out from Micronycteris. It has a broad range of diet, whereas this neighbor, the Macrophyllum, has a much tighter, smaller diet. And of course, way on the big scale, you see that Vampirum is the only bat large enough to eat those large prey items. So now we're going to move from figure illustrations, where it's just one item and one story in a single figure, to illustrated books. This is where you get the luxury, if there's grant money, if there's a possibility for a bigger project, to tell a larger story, and in this case with illustrations, so you can reduce the text and expand the amount of visual. The first one I'm going to show is a project that took place in a conservation area in Guanacaste, Costa Rica. The researcher who had been researching there as her principal site for some time said, I know that this area, this nature reserve conservation area, is surrounded by a huge number of schools. And I'd like to get some message out to them of what's going on here. Well, when I got to the project, I sat in the room with all the people that do tours of that area for visitors, for the students in the area, for tourists. They said, oh, great, you brought an artist with you, a scientific illustrator, I'm pretty sure was what they said. Todo la conversación está en español. So we're talking in Spanish now, and they're saying, oh, great, can you help us do some nicer coatis? Those are the animals that are pictured in the lower part of that image. Could you add some animals or plants in our games and text images that we give to the kids? And I said, well, that's not my goal. My goal is that I'm a scientist who also does art, and I would like to facilitate telling some interesting stories about what you all do here in a very strong fashion, in an illustrated way that will attract the kids, and hopefully they'll bring that book home to their parents and they'll all read it together. So then the goal is to educate the local community. Now we have a map that's showing, you can't see these. If you look squint, maybe you can see that there's tiny little flags that show all the schools. And there are 46 education centers and about 2,000 students. So this is a huge population that surround this conservation area that probably know very little about what goes on there. So instead of having some games, instead of having just text, I wanted to show them visually and explain some of those stories. So a few of the pages focus on fascinating stories. In this case, one of these slides on the left is talking about amazing mutualism between an ant species and a plant that has evolved thorns that are designed through time and evolution, through natural selection, to allow a place where that ant can enter and raise its brood. Well, in turn, the plant gets something very special. That's the home of the ant now. It wants to protect that area. It's being fed by the plant even. So everything is there. That's its home and food. So if there's anything that comes near that plant, those ants are there to defend it with powerful stings. And they can even fight off, in some cases, giraffes in Africa in this type of a system. But here they even pick off vines that might try to cover the plant and stop it. And then on the right, a fascinating story, because there's a spider, the first vegetarian spider noted, because it's using that plant, the food that the plant is creating for the ants, and it's hopping in, grabbing some food before the ants can get it. And it's the main resource of that spider now. It's eating almost exclusively this vegetarian resource, high in protein, but still created by a plant. And one more thing I wanted to do for those families was try to dispel some of the myths that are common in a tropical region, especially in Latin America, that all snakes are dangerous and all bats want to suck your blood. In both cases, it's not true. Of course, there's many snakes like boas that are very beneficial to the people that live there. They might not know that. The machete might come out whenever they see any snake and they'll cut the head off, assuming it's going to hurt them with venom. Likewise, with the bats, they might be quite happy to kill any bat that comes near their home, thinking that it's going to suck their blood and give them rabies. Not true. Again, lots and lots of bats, a high proportion, are insectivores. They're flying through those night skies and eating up mosquitoes by the millions. And that means not just that they're getting rid of a pesky insect, they're getting rid of a vector for dangerous diseases, especially for their children, malaria and dengue and new things like oropuche and chikungunya. There's so many diseases that are being vectored by mosquitoes right now on our planet. This is just a simple example that I want to share as a reminder that as an illustrator, if you want to reach a broad audience, if you want to inform them more about the science, illustration has one of the tools that's very powerful is zooming in. It's very difficult to do with a photo that you can mostly do with macros other than with very great and expensive equipment to go down deeper. In this case, the scientific researcher and evolutionary ecologist Egbert Lee, wanted to tell a story about sponges for his particular reasons in the story. But to do that, he said they have to know how a sponge works. So my job was simply to show something about what his text was showing. And that text was very complicated. So I wanted to illustrate it in a way that they could understand that if you went into a sponge, basically it's like a big straw. It's just sucking all the water on the outside of the sponge and passing it through towards the center. En route, it's grabbing all the little particles of food that it wants to eat and passing that water on. So if you look on the far left, all those little flagellum are waving, 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 and moving the water through the sponge. All right, now we're going to move to another illustrated book. This is a book that is about both bats and frogs. It's a story that involves them both, but I wanted to take the opportunity to tell two stories. Just a general story about bats, what makes a bat, what's important. And I wanted to do it for a very general audience. I wanted to make it fun. I wanted to make it very interesting. Well, if you want to tell a story about bats, you need to tell at least two stories. You need to tell the story about the evolution of flight. Flight is critical to what makes a bat. And you also need to talk about echolocation beyond anything else you want to talk about. In this case, I wanted to make it interesting by including some text that's in thought bubbles from the organisms. And as I was creating this slide, I realized I didn't take the chance to give a thought bubble to that poor pterosaur who evolved long after insects did. But the story is basically that bats were the last things on this planet to evolve the ability to fly, and they're night flyers. So they have a new niche for them. They can look for their food source at night. In contrast to that black and white world and grays of the bats, I wanted to tell the frog story as well. So I did that in all the bright colors of frogs. Frogs can be very brightly colored if they're venomous and have a bad taste. So they advertise to the world of predators around them, don't eat me, I taste horrible. My colors tell you that as a reminder. And I took the chance to talk about female choice, just to remind people that in this system, the females are the ones who are choosing who their partners will be. The males are calling all night, hoping to find one. The females choose which one they like. And now I just want to show you the book in person. So we have our book. It's a story about frogs from one direction. You flip it over, open the book from the other way, it's a story about the bats. All right. So as I said, the world of bats is all in grayscales. We're opening those pages. We're moving through the stories, and we get to a center page. Well, before we get to that center page, let's go back to the frogs again. We're going through that colorful world of the frogs. And when we get to the center, we find out that there's a very different story than that the reader might have expected. It is a question about frog calls. Are they a call to find a mate? Or perhaps that call is actually advertising to a bat to eat the frog. So is it a dinner call or is it a mating call? And in this case, I wanted to make a fabulous spread. So the inner page, the inner centerfold opens up to the big scale. And I'd be happy to talk more about what I intended to use this for later if we have any time. All right, so very quickly, this was a project that was designed to do some community science and create great fun for general public to talk with fireflies. A firefly communicator that gives the light signals species specifically to the fireflies, which really should be called lightning beetles. They have nothing to do with the fly around the world. My job was to create a story that would make you excited to use that tool and how to use it. So take you out of your urban setting. It's nighttime, it's dusk. What a perfect time to go see if there's some fireflies out there. You grab your firefly communicator and out you go. And you go to your local setting. In this case, of course, luckily it's nearby. They can walk to it. And they go and they see, wow, they can see those fireflies glowing. They sit down, they're patient, and now they have their wow page to advertise their product. That yes, if you take the time and you're patient, the fireflies will actually come to visit you because those males flying around will think that that signal you're giving out is a female saying, please come to visit me. I want to be your mate. All right, so the last illustrated thing I'm going to show is one of my favorite and probably the most effective illustrated item that I've created. This is for a book about the greatest mammal migration on the planet. And it is about 5 to 10 million bats that are moving from one part of Africa. We're on the continent of Africa. They're moving from the west to the country of Zambia, farther down southeast. Within Zambia, they're moving to a tiny little national park called Kasanka. And within that park, those 5 to 10 million bats are all going to a small swamp forest that is filled with fruiting trees, and they're gobbling up 330,000 tons of fruit. And they're also dropping those seeds as they fly and planting seeds along the way, planting trees along the way. So they are ecosystem service. Here we are. These are the local residents in Zambia in the national park. And here's what I'm going to provide for them. We knew that there was going to be very great difficulty in what they could print, what could be afforded by this community. So they wanted to be able to provide something. And I said, let's do something that will be printed on one single sheet of paper. And from that single sheet of paper, they will be able to do the work. The people from the community, and these three guys are actually guides, I believe, in the park. They're trimming off the edges, making one cut down the middle, gluing those two large pieces together, folding it, and they have themselves an accordion book. So let me show you the accordion book. Tiny. Fits, as they like to say, right in their pocket so they can carry it with them at all times. You open it up. All the guides have this with them, they say. They give it out to the local community, the tourists that visit. It has all the information about those bats. It also has an added wonderful effect of just being fun because on the other side is the bat with its pop-up head. All created by them with a single sheet of paper. And then because you have a system where you want to really show the reality of things, that is the full-sized bat, actual size in a large version. All right. All right, so penultimate item, murals. Murals are just something I love to do. On the right side of this, the wall that you can't see very well, I did it with the community of Gamboa along the Panama Canal in Panama. And we used a mola theme from the local indigenous people called the Kuna, their style of art. And after I was done with that community project, I wanted to do something for me. So I created an anteater head, which is also a nest for the leaf cutters. As you can see, the green things moving in, those are all cut leaves that they're moving down into the nest. And the queen is actually the eye of that anteater, if you could look close. And above it is a giant anteater, a giant leaf cutter ant that now is actually just cutting through the building itself. Just something fun for me to do. All murals are fun for me to do, but this one has the specific point of guiding someone into an amphibian rescue center. Why is there an amphibian rescue center on our planet? Why does the Smithsonian invest money in such a thing? Because about 1/3 of the species of frogs on our planet are in decline and are an endangered list. So they're highly threatened. And if you can imagine, there are a number of species of frogs that are now living probably only inside of rescue centers, and with the hope that someday they will be reintroduced back to the real world when we have figured out how to deal with a fungus that is killing these amphibians. Another project I had with murals was to share the importance of mangroves in their local community. So this is a school on the coast of Panama. On that same coast of Panama where this school is located, their neighbors are now building skyscrapers, investing millions of dollars, and they don't want mangroves in their community. So they build a cement wall, and now those cement walls shunt all of the water that would have gone into their section of the mangrove, and is pushing that water into the smaller existing mangroves and flooding local communities. And if you want to see anything about my murals, I have uploaded a lot of different murals in a time-lapse fashion, so you can see the process going from white wall to something hopefully beautiful. And if you just put in YouTube on a search, just put in Dakilo designs and murals, those things should come up with primarily my work. And the last one, you've already seen this slide because I used it as a figure. That same researcher said, "Oh, I'd really love to have that figure on my wall as an education tool. I work at the Smithsonian Tropical Research Institute. We have lots of donors that come here. We have lots of visiting scientists. I can use that image to tell all the stories that I want to about my group of bats." And so that's what we did. All right, now we move into something completely different, and something that was the biggest challenge for me in my illustration career, I would say. This actually was a project that was offered to me by Barrett's twin brother, Arnold Klein, from the Child Mind Institute, a project called Mind Logger, in which they want to make a mental health app for phones. Why was I invited to help as an illustrator? Because they clearly recognize that if you're going to do a mental health survey, a self-survey, and you need to answer questions day after day, and it's only text, it's going to get so boring that a lot of people are probably not going to answer correctly. They're not just going to even think about it. They probably are going to stop doing the testing, everything about that. So why not make it visually exciting? Not just because it looks better, but also because it can reduce the time necessary. The picture will remind them of what the text is, and they won't have to read the question each time. They'll just say, "Oh yes, that sloth hanging on a tree reminds me that this is about activity level." So if you look at these images, each of them represents something about activity level. Now they don't have any text yet. Take a look at take a look at the far left, and you see a foot moving up and down. Take a guess, get it in your head what you might imagine that is. Look at the next one with this hand. And
Maybe in the chat.
you can even share your idea in the chat if you want.
Yeah, before we do the big reveal.
So take 1 minute to look at any of these items and write down what you think any of them might mean. Something about an activity level and
Oh, I see.
restlessness.
Good.
Very good.
Pacing. Me right now.
Indeed.
That's great.
So let's take a look at what
Fidgeting.
fidgeting. Excellent. So we've got one, a direct hit. We need to go back
Oh,
to
sure. Just there we go.
There we go. So now you can see what I was hoping to do for these particular items that needed to be on the mental health survey. Now, all of you are looking at them, and hopefully most of you are thinking, yeah, that makes some sense. But of course, this is why I say this stretched my abilities, because it was very difficult in many cases to do an image that would capture, in some cases, very complicated emotions or concepts. Something that I didn't mention is that there's a worry with this app that if I'm presenting a human face with a particular emotion, that might affect the user in some way, like, oh, I see a really grumpy face, that's making me feel grumpy. So in some cases, I thought, why not use animals? And both Arno and I thought this was a fun idea. Take the person outside of their own personal space, and maybe they'll be able to see these images, have fun with the images, and not be as affected by that input. And I just wanted to share this because we're doing a visualization challenge with these emojis at the end here in the final hour. It'll be one of the things that we're going to do. And so I wanted to explain to you some of the problems that I had, especially in this one. It shows that I'm trying to talk about speech quality, but without being able to use words. I needed to use only imagery again. So there's one here that does not have the title. And please write down in the chat if you have any concept of what that individual is doing in terms of their speech quality.
Oh, speaks fast. That's interesting. Singing. Freedom of talk.
Many of you have said something about speed, so that tells me that it was unfortunate for me to choose a hummingbird. I should have chosen a bird that we don't interpret as just being fast, but just any bird that
Descriptive.
Concise. Okay, so let's look and see what we have. All right, so some of you said concise, very close to speaking coherently. You know, somebody who is capable of saying clearly what they want to say. So great. Remember, you're going to have to use that same thinking if you choose this as a visualization challenge in the last hour. Oops. All right, so I'm going to finish just by saying that for me, making art and using the natural world it's just part of who I am. I didn't choose this because I thought it was a great job. I chose it because it's what I want to do, and I've had the luxury to be able to do that through grants of the National Science Foundation and other forms. And the other thing I love to do is collaborate with Barrett, and that's why we're both here talking to you today.
Well, now we have the opportunity for a tea break of 3.5 minutes. And immediately after that 3.5 minutes, Damon and I are going to jump into some enthralling visualization challenges, as well as opportunities to share additional items that we haven't shown or discussed in the previous hour. So feel free to take a few minutes. What I'll do is keep should I do the Q&A? I'll open up the Q&A and I'll be right here and Damon will be right here if you'd like to ask us questions. Here are a few already. Great. Well, let's check them out. Meanwhile, everybody else, drink your tea or beverage of choice for the next few minutes, and Damon and I will walk through a few of these questions. And now Sheen has the question, which software is used to make these figures for illustrations? And Damon and I have some overlap, although Damon focuses more on much of his work with vector-based, for example, Adobe Illustrator, than I do. Do you want to?
Yeah, I want to answer that by saying that the reason that I use vector-based tools is simply because I want to be able to manipulate what I create in many ways. When I'm collaborating with a scientist, that scientist might give me a reasonably good concept of what is desired, but it almost always is going to include lots of changes through the process. So using a vector-based tools like Illustrator, I can quickly make changes on the fly, even when we're just talking and say, how about this? Colors, of course, are no problem. You can move from color to grayscale. It is a powerful tool beyond my comprehension when I started 30 years using it.
And as we've mentioned in previous workshops, there are freely available programs, surrogates for these expensive programs like Adobe Photoshop and Illustrator. For example, GIMP, Krita, and for vector-based, you've got the Inkscape.
Yes.
The next question has to do with this guy, the Firefly, right? Designed by Joey Stein. And what does the Firefly necklace's button do when pressed? Well, I've got an Android phone, but if you place this on,
I don't know if the battery works in that right now.
Oh, let's see.
Let's see.
Oh yeah. Oh. So ding, 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 ding. So you can either manually press this to light up and attract male fireflies, or you can place it on an iPhone and program it for species-specific bioluminescent calls. What do you use to make your illustration? What did Barrett and Damon enjoy the most about working with other people to make illustrations? And what do they find most difficult?
You go first.
I love collaborating on exciting projects. I only have one life as far as I know, and I've chosen a bunch of things that I find really exhilarating. And when I find other people delving in realms that are really intriguing to me, and if I can be of any service, for example, to render their works more broadly accessible, that's exciting to me. So combining the clarity, the accuracy, as well as the aesthetic bonuses to make things work, to reach audiences with messages that are important to me, that's really special.
And for me, maybe one of the most important elements of collaboration is that I'm working with people, scientists, who do totally different work than I did. And any scientist on the planet is somebody who wants to understand more about the world. That's what a scientist is. They want to learn more about. And so by working with various collaborators, I'm learning about all kinds of things I never had any idea about. I'm illustrating about grasshoppers getting infected by metarizing. That was definitely not one of the topics that I studied. Bats were just flying creatures before I got to the Smithsonian Tropical Research Institute, and they are very fascinating. I've had the luxury to see them in person. It's just, yeah. It's
That's wonderful. Right. It's another way to learn, to expand your horizons.
They also want to know about what was most difficult.
Yeah, so this is really interesting. What are the key things to think about when creating illustrations for scientists versus the public?
Well, okay. Who is the general public? It's all of us. Even though I am a scientist, when I'm doing a project about somebody else's science, it's unknown to me primarily. So I need to understand that, and I'm doing it as a general audience. Yes, I have a scientific language, so when we're talking, I'm thinking in a similar language, and I have to think about how to change that language for the general audience. But In either case, you want to be clear and concise, and then hopefully you have the luxury to also be beautiful in what you're presenting.
This is a question for Damon. Of all the media he uses, if he has to choose only one, which one will it be? And I assume formats, murals,
Oh,
or
yeah.
booklets versus.
Well, my favorite at this point in my life are the tiny little books like The Greatest Mammal Migration, done for the Max Planck Institute. It is a great little tool. I had no greater thank you than what I received when they shared the stories of how those kids were keeping a copy of that book in their pocket so that they would be able to share it with everyone who came into the park. Now that was fabulous.
And then Maria asked, OMG, can I buy it? So some people are interested in some of the works on display. Do you sell them? Are they available?
It's not really what I do. Of course, you can contact me. I think, how would they have my contact, any?
We'll make contacts available through
Yeah.
Daria and Joanna
So
on the site.
you could personally contact me if there was some particular item you wanted to get your hands on, so we could see if that's possible. Yeah, sure.
Disha asks, also, it would be great to hear Damon's thought on how to improve communications for audience with any disability, for instance, visual impairment.
Oh, wow.
It's a tricky one.
That's a fascinating question. And of course, both Barrett and I are visual artists. Even to imagine trying to do what I do without that ability is beyond what I can comprehend, really. I don't know what I would do to enhance the life of visually impaired with what I do, except that you would collaborate with somebody else who has that capacity to take what I'm doing and they can alter it in a fashion that can be read by Braille. It could be interpreted by audio for the stories. Yes, I'm doing some collaborations with people who have turned some of the books that I've done, illustrated books, into videos. Again, it's a visual item, but it has sound quality to it, not just reading.
Here's an example by Joey Stein working with David McCornack, in which they molded and then cast sculptures of the hearing apparatus as well as the music apparatus produced by and exhibited by Crickets. So this is very much a visual, but also a tactile means of learning about and appreciating the sound production and reception of our first musicians.
And of course, museums are the place for anyone who's visually impaired, especially those that are designed for touching and feeling, where they can actually get in there and feel what an organism is like.
Yeah, so some works that can be displayed in museums, like this housing of RNA in an HIV virus, some things are tactile, as Damon is saying, at museums, and those can be really enriching for visually impaired audience or participants.
Just the opposite of Fine Arts Museum, where you can't even get close to the item.
Except for the token occasional piece. Are you using the same publisher for all your books, or are they printed locally to your research areas?
There is no publisher, is what I can tell you. The answer is to that. In general, there's no specific publisher because each work is so different, and I'm attempting to make things that are actually self-publishable, that you can print yourself. Especially the pinnacle of what I've done was that bat book about the greatest mammal migration, because it is literally just an 8.5 by 11 sheet of paper, a standard sheet of paper, printed on both sides and turned into a book.
And I'm going to end with one more question so we can jump into the next phase. And that's, how much of your work comes through, this is from Hal, how much of your work comes through personal connections versus cold calls or the like? It's wonderful how much collaboration you've been able to do, and I'm wondering about how these relationships formed.
I can tell that story for me in a very simple way. That those relationships formed during that long period of time where I was a researcher living in a very unique place. The Smithsonian Tropical Research Institute, located in Panama, is an international setting. It is also a place where all the scientists are working together, all types of scientists working together. And those connections that I've made there have continued throughout my life. And I am sorry to say, but I don't even have a working website at the moment to show what I do. All my work comes through word of mouth and working with people, and people seeing those figures in publications say, I need to have some work done as well. Can I get that contact? I'm not making an effort to make a lot of work for myself. It's happening enough as it is in general. And I should make a little more effort to broaden that scope, but that's my.
And for me, it depends on what stage or phase of life professionally I've been in. For example, years ago, I was a freelance scientific illustrator. So I'd try to get the word out by word of mouth. I'd put postings, and today I'd use social media, for example, and the word would spread. Then when I worked at the American Museum of Natural History, I would do freelance work, and that was easier. That came to me. They wanted to find out who was producing these exhibits at this world-class institution. So then I'd collaboratively produce exhibits for the Bronx Zoo and other places. And now as a professor, people see, oh yeah, he still does scientific illustrations and all this. And through conferences, through meetings, this is where collaborations can form. So I might have a poster, I might give a talk. And then when we perform a scientific collaboration, that can translate into a scientific visualization collaboration in everything we do.
And we have to recognize that the academic scientific world is a very different place than many things. It's a small community. It's a small niche community in terms of you really do meet a lot of the people that share your interest in science through various methods like meetings, reading publications, making contacts. It's very different from somebody who would be an illustrator in the general world trying to get themselves noted as being somehow better than some other illustrator.
And before we move on, I wanted to share one example. This is one of the 4 case studies where I hired someone who happened to be my mother. And this is the Tree of Sleep image. So you see it's quite large. And when I zoom in there, you'll see the details of some of the work.
Yeah, go up with it. Yeah, that's good.
And we can share more examples, but I wanted to jump right into, oh my goodness, hold on, I need to plug this thing in. Damon, entertain.
Quickly. I'll show you a fun item. This is another little book. Whoop, wrong way. It's all about katydids that have evolved to look like leaves. So I'm not going to take the time to go through all the little pages, but the punchline is quite fun. This katydid has now believed, finally, I look just like the vegetation. No more worries, except a mantis that's pretending to be an orchid flower is waiting to consume that poor guy.
Okay, now that our 3.5-minute tea break is over, we're going to move on to 3. Get a load of this. 3 visualization challenges.
Options.
Now, you choose just one of these. I'm going to present the first. Damon will briefly present options 2 or 3. And what we'd like for you to do is in the next 20 minutes, create a visual concept piece related to either visualization challenge number 1, 2, or 3. Mine is an octopus's dream. Not the octopus itself dreaming, but what would the dream of an octopus look like? And maybe some of you are familiar with this book, When Animals Dream, and who's on the cover but an octopus. And maybe some of you are familiar with this video that famously, relatively recently shows an octopus with the chromatophores going haywire during sleep, showing not only color, contrast, but texture differences across the cephalopod's body. And as you'll see, it's blanched white and then flickers and pulses with twitching as if it's in rapid eye movement sleep. And you get chromatophore changes in patterns across the body, which even suggests, at least to some, that maybe the octopus is dreaming about something specific and predictable. That's your visualization challenge 1. What is that octopus dreaming? Number 2.
All right. So the last thing I talked about were these mental health emojis. And as I said, some of the things were very challenging for me. And some of them were challenging because there's almost like a time component within the activity, within the emoji. So yes, it might be easy to do an emoji of worry, just having a face that's worry. But no, in this case, the question is to the self-assessment is, are you trying to avoid a worrying activity?
Cool. And number 3, if you so choose.
So this is a scientific area that's very interesting right now. There is climate change on the planet. CO2 is increasing, and there's an insect decline. And they seem to have some relationship. So this is called the nutrient dilution hypothesis. The concept is that soil nutrients really aren't changing in any great way, but plant biomass is increasing dramatically with the new amounts of CO2 in the atmosphere. That means that the nutrients in any given leaf have been going down. What does that mean for an herbivore? Something that only eats plant material? Well, it probably is suffering, and there's good evidence to show that indeed it is playing some role in the decline of insects on our planet, that these herbivores are eating, but not really growing in the normal manner.
Okay, so brief review. Number 3, the nutrient dilution hypothesis, with these 3 factors in mind and a result that herbivores are suffering. Visualization challenge 2, an emoji showing the avoidance of a worrying activity. And 3, what would it look like if you were inside an octopus's brain and you're dreaming? Okay, so I'm going to stop sharing. And it's time, while you're working on that, for the next 20 minutes. Daria or Joanna, can you put up our pop poll for options for those who might want to be educated, entertained simultaneously? Oh, there it is. Would you like, during this 20 minutes plus, would you like for us to tell more natural history tales or stories related to the work? Would you like for us to show tangible, that's IBLE, examples of art? Or an additional example? I showed 4 stepwise examples before. Do you want to see a 5th of mine that I deleted from the presentation? Would you like to continue the Q&A, or would you like dead silence?
Working in dead silence.
Dead silence. I mean, you can always mute us, but have you just had enough of our voices? Do you want dead air, dead silence, while you continue in peace, finally, on your visualization challenge 1, 2, or 3? So we've got 2% really wants that dead silence.
And we know that
the numbers are growing.
And we know that 2% is, that's
it's 4%. The numbers really are growing.
They're growing.
And 3%. Well, okay.
And only 6% of you want to hear us talk about Q&A. So I guess you don't really want to hear us talk. You want to see things.
Well,
that's good. That's good.
I guess our continuing to talk is abiding by the selection of the masses, because it's still down to 4%, 7% Q&A, 8% additional Barrett example, 36% tangible examples of art, and in first place with 68 out of 155, 69 out of 155, more natural history tales. Let's tell them some tales. I'm going to end the poll now because I don't think silence will overtake the more natural history tales at this point. Oops. Thank you for taking part in the pop poll. Now, let's pick a tale. Let's pick a tale.
Give him an insect example.
I'll give him an insect example. So I mentioned that one of the products that we worked on was the Insect Epiphany, and that I was working with my mother in part. Well, she produced well, actually, I'll show the contents page. Here it is. And you see there are her beautiful insects all around. And here's the original where I separated those out and played around with their appearing in the different parts of the contents page. I'll choose an anecdote. Let's see. Here's one. A pompilid wasp, a tarantula hawk wasp. Pepsis is the genus. And if you were to go to the Southwest deserts, like Arizona, you can find one of these glamorous, glorious, orange-winged and orange antennae-tipped wasps that will very gracefully and brutally sting a tarantula, a massive, hairy, theraphosid spider in an appropriate location to render that spider paralyzed. Will grab then and drag that massive arachnid to a pre-dug burrow, stuff it in there, lay an egg, and the developing larva will feed on the still living paralyzed body of that Pepsis wasp.
What a gory example you've chosen.
Your turn, David.
Now, I want to tell you another thing that I do with my books, and that is to try to share some kind of public awareness, especially about things that are going on the planet. So, of course, this is one of the things that I share about. I talk about global climate change. And although we don't talk about it very much, one of the easiest methods to deal with climate change is to grow more forest rather than cut it down. So with this, 5 to 10 million bats that are all eating fruits of trees and they're pooping them out, they are ecosystem resources. They are planting trees about everywhere they go, and they're acting as a free source, 5 to 10 million workers planting trees on our planet.
Well, Damon did a number of the illustrations in Egbert Lee's book that he mentioned, and I had an opportunity to help out with a couple of them, and they might be difficult to see because they're small in their representation. There's one.
Well, that one's going to be the toughest.
And then there's a second one there.
That's good.
And in the first one, the reason why I share the first one is because the leader of our third workshop in the workshop series, Danielle Van Brabant, produced this with a little bit of my guidance, and it involved the evolution of the segmentation of arthropods. So how can you show that this part of one arthropod is the abdomen, but this section of the body of another arthropod is the abdomen? And she did so elegantly with colors. The second one was a collaboration between my mom and me, and it's called the homology of insect mouthparts. So whether you choose a beetle, a true bug, a fly, a honeybee, a moth, you can look at the radiation and diversification of different insects based on how they feed. So for example, a lepidopteran, a moth, for example, will unfurl a proboscis and extend it usually down the corolla of a plant to feed on the nectar, while an insect with more primitive mouthparts would have transversely chomping mandibles, and list goes on. Great example is the synapomorphy, the shared derived character that defines true bugs. They have a piercing, sucking mouth part to draw either plant juices or animal juices.
Same book. This is a story, a natural history story about a concept at the beginning turning into something very different at the end. The author, evolutionary ecologist Egbert Lee, working in conjunction with Christian Ziegler, famous National Geographic photographer. The concept was that this would be a coffee table book in which each and every page would have a beautiful illustration or a beautiful photo alongside of that text so that it'd be really fun to read. What happens in the process? Publisher says that's not going to work for us. It's going to be too costly. You go from my job as being the illustrator for very tricky scientific concepts about evolution. First thing that happened was, well, we're not going to be able to do it in color. That's too expensive. Can you change all of your illustrations that you've done from color to grayscale?
Sure.
Sure, I'll just do it with a click of that button. Oh, that says convert to grayscale. Click. Oh, it didn't quite work out like I hoped because I used a red next to a green and an orange, which showed up very beautifully in color, but they all are the same grayscale. So I had to go through each and every one of those illustrations. Yes, it is a vector tool. Yes, I could use methods to make it more simple than having to change everything like Barrett would have to do with his mother's beautifully painted and drawn images. But it took time. And what a sad moment to know that your spectacular illustrations are going to just be a little grayscale thing next to mostly text.
Here's one from the Insect Epiphany. I decided I wanted to talk about in my insect products and how they've affected human culture. I wanted to choose a strange example where you might not expect insects to play a big role. My sister is a luthier. She makes instruments. And she never thought getting into the music business and instrument making business that her life would be as a wannabe ancillary entomologist, but indeed insects play very clear roles in some cases with her instrument production. So in the book, I created a figure that shows a viola that she made. And then you see that there are pointers to various insects or insect products that surround that viola. So for example, in one corner, you've got a honeybee colony, and the production of beeswax contributes to viola in a few different ways. The honeybees also affect even the naming of parts of the viola, because those pointy parts on the tips there are called bee stings. And propolis can be used on parts of the instrument as well. Propolis is tree resins that are combined with honeybee products to seal up, and they have antimicrobial agents oftentimes too, so to remedy or prevent pathogens from spreading, but also to seal up a nest can also be used to seal up the interior of a wooden musical instrument. And then the very color in the varnish of violins and violas can be based on these scale bugs, cochineal bugs, that feed on Opuntia prickly pear cacti. And then on the backs of instruments and in other areas, you can have shellac or lac from lac insects. So a variety of insects can be involved in positive ways in the production of an instrument. And I wanted to convey that in a figure that included the insects surrounding that instrument.
When I was given the option to do this Firefly book, I said, well, yes, I want it to be fun. Of course, I want it to be fun. That's the goal, to use this device to go out. But I also want to tell them about the most important aspect of the organism, that they use bioluminescence in order to communicate. I think the reader should know something about how that works. So I wanted to make this simple little thing at the end of the book that would explain how an organism generates the ability to create light. and use that light in very specific ways. The ability to make a pattern with light, very fascinating. The metabolism and chemistry involved to do that.
Right, I just pulled this from the wall. These are thermal images of honeybees. So for example, that's a single honeybee. And you see the midsection of the body, the allotrunk or mesosoma, is piping hot. So a honeybee can contract her wing muscles without flapping her wings, generating heat for various functional reasons that help with development of brood, that can just warm up fellow members of the colony. And here you see on that top image, a whole nest with several parallel combs dangling. And you see the hot center is the brood comb on one of these. I'll point that out. So the yellows down here, this would be on the order of 95 plus degrees Fahrenheit, because that's 35 centigrade, something like that, so that you can properly develop and maintain a temperature conducive to survival and in a honeybee colony. So thermal imaging is one way that I otherwise convey the invisible that tells stories visually.
This little thing, this is a tree. If you can see it at all, you might notice that all those leaves are bats. Why was this made? This thing was originally made for Biotropica as a cover art, but it later became something important because the maven of bat research, Elizabeth Kelko, had an untimely demise, dying at a research site. And she had so many people who had learned from her and followed. They wanted to have a memorial service for her. So I created this tree and recreated it in the form of all bats instead of all wasps, which was its original. And it includes 50 different organisms within the tree, tropical organisms. So if you had the time and the patience, you might be able to find 50 species of animals that live in a new world tropical rainforest.
And my latest Halloween mask, this is an example of visualizing, well, wasps and the nests they create. So wasp architecture. So it's not actually a nest, and I can show you real nests behind me, but I used paper mache from those wasp nests and created basically a brood comb out of paper straws used as bee hotel components. And then I just inserted branches to give the suggestion that this is an arboreal wasp nest. Dolica vespula maculata, for example.
Dun, dun, dun. Oh, there's a real nest. So I pull paper from an abandoned nest to create this new nest head. Is my insect epiphany book that has been eaten by termites. Another way to visualize my book, I'm feeding it to different insects, but I'm also letting honeybees build a comb on my insect epiphany book, collaborating with non-human animals.
And don't tell the publisher that those advanced copies are being eaten by termites.
No, they love it. And here's another example of my mom's work for the sleep book. So here are 4 ducks in a row, right? 4 mallard ducks. And the intriguing story here is that from Niels Rottenbor's work, he showed that, well, if you go out and you look at birds and you think these birds might be asleep, say, for example, with their bill resting on the side and eyes seem to be closed, look at a bunch of ducks. Next time you see a bunch of ducks, you'll see that when they look asleep, they tend to have both eyes closed, save for those that are on the edge. The eye facing out tends to be open. So what did he do? He experimentally addressed this observation by placing 4 ducks in a row, and the one on one side and the other end typically exhibited what's called unihemispheric sleep, where one hemisphere was showing high frequency, low amplitude waves suggestive of a wakeful state brain, with the contralateral eye open, and the other hemisphere had high amplitude, low frequency waves, and the contralateral eye was closed. So sleeping with only one eye open at a time. And it's hard to see here, but the 2 in the center have both eyes closed, and the 2 on either end, the one on either end, has that eye facing out open, to show that idea that there's probably some predator avoidance going on behaviorally, thanks to unihemispheric sleep in ducks. Okay, so I think we should wrap up. We've got enough that we can spend at least a little bit of time on each, and that's what I'd like to try to do. I think we can guess which visualization challenge this is. What does an octopus dream? And sure enough, we have no clue what's happening in another human's brain, for the most part, let alone in an octopus's brain. So does that octopus see nothing? Next, challenge number 2, avoiding or running away from worry. Damon, do you want to tackle this one?
Well, and indeed, there's some other people that I've seen already that avoiding a worrying activity did something very similar to what I did. Yes, I did a lot of emojis that had to just do an image of a face that would show an emotion. But this one was complex in the sense that it's avoiding the worrying activity. So somehow the worry has to be separated from the concept of avoiding that worry.
How are you avoiding or escaping? And this is an adorable,
Yes,
concerned individual.
it certainly is.
Okay.
This is the challenge for the
Number
for the world
3.
of herbivores. Indeed, I mentioned an insect herbivore, but of course,
Look at the ribs
yes.
on that ungulate.
So this poor deer eating. All the humans around it are probably unhappy if it's gone into their gardens. But yes, a good use of the effect.
Wow, I love it because you still see the leaves, and even giant leaves, in the presence of this ungulate herbivore, yet emaciated, ribs showing. I think that's beautifully, effectively done.
Yeah. That's a great first step for that one.
Oh,
Yeah.
I bet this is the octopus dream.
Indeed.
Oh, that's beautiful. I love it.
Yes. What do we know about octopus eyes and color vision?
Well, cuttlefish vision, another cephalopod, is kind of this mixed mystery as far as I understand. Roger Hanlon's group has looked at cephalopods which presumably are colorblind, yet they perfectly color match their surroundings.
Yeah.
So do they have visual components, photoreceptors dispersed across their bodies that they're responding to? But in this case, yeah, that's a really good point. Are they seeing in their dreams with the organs we call eyes?
And would any organism dream in color if they couldn't visualize color?
Right. But I love this mix of water and the suggestive surreal components. Really,
Yes.
really beautiful work.
Very beautiful.
And in the chat, if you wouldn't mind telling us who you are, who produced this octopus dream, that'd be great.
Beautiful.
Oh, there we go. That's literal.
Now that is a beautiful example.
Get away from that worry.
I'm gonna tell you, you can take over my job right now.
You're hired.
You're hired. Yeah, that is fantastic. Yes.
Beautifully done. That tells the story.
Yeah.
In high drama form.
Very, very well done.
And I love the color outline. It really adds to it.
Yeah, there's a lot of aspects to that, why it's effective. Indeed. That exclamation point separating the running from the other image. Yeah. It's good.
And if I could read your signature, I'd congratulate you by name. Eva, was on. Okay. Next one. Oh, looky here. Sure enough, you've got the classic sleeping octopus.
Oh, right. On
Yeah.
a pillow.
Yeah. Oh my gosh.
Nice setting.
In bed. Now,
And-
is this octopus dreaming that a cephalopod's rightful place is to sleep in a human-style bed with pillow?
And a bird?
And a bird.
Is that cephalopod imagining that besides all the amazing things it is capable of doing, that it also wants to fly?
Love it. Love it. Oh, this is number 3.
Oh, nice. Again, really, okay. You've used some graphics for the content of what's going on with the leaf nutrient level going down. Very good, very clear. And our 2 different sides. So we are looking at a world in which you can see one side where the content of the nutrients is higher than others. So there's a difference in how those 2 sheep are existing. That's very well done. Very
And,
clear and concise.
and it reminds me of the old Mad magazines where you have the fold-ins.
Yeah, and I like that you've indicated that it's it almost looks like it could be a spread across a page. So you have 2
Yeah.
pages.
Yeah.
Split down the middle. Yeah.
Well done. Oh, same, number 3.
Again, very good. Trying to indicate what is going on with pop-ups, with magnifications of what's going on within a leaf.
And what I like here is you say soil consistency. Maybe you don't even need to say it. It's nice to have the text complementing what looks to me like the same density of symbolically colored elements
Right.
that would suggest soil nutrient availability.
Yeah.
And then the density as well. I mean, the number of plants per unit area.
It's interesting. You've captured
And
a
look
lot
at the
of
drooping ears. That's great.
Yes, if one could capture a bunny looking rather sad and tired,
Yeah.
that would be a very effective method. Yes.
And the symbolic increase in CO2 on the right side. That's really great.
Yeah.
Oh,
Wow.
yep.
Very succinct.
Get out of there.
Right. In using the same style that I showed you, that it has to exist within a box because this is going to be on a phone app. So perfect that you have used the space that would have been available to you. You put the avoiding of the worry. The only thing that I'll show you in mine that I did differently is that there was worry about a particular activity. But yes, still avoiding worry.
Number 3 is a popular one. This is great.
Yeah.
Nutrient dilution hypothesis with the 3 components and the results listed. And here we've got it visually.
And beautifully drawn as well.
Yeah.
Oh, I like the half full.
Yes, the half full
Deer or cow almost. I don't know. From this size, hard to tell. But anyways, half filled herbivore.
Uh-huh.
And with high nutrients level to the biomass versus low level of nutrients to biomass.
That's really nice.
Very good of using the half full dark light contrast. Yeah.
Nice.
Ooh, nice.
Nicely done. Aha. Get away from the left and go over there to the sunshine and heart eyes on the right.
Oh, that's interesting that that by avoiding a worrying activity, they're happy.
Yeah, that's right.
Right. So yes, you're going beyond what the question even is and saying that the need to avoid the worrying activity makes you happy.
It's not just relief. It's euphoria.
Yeah. Again, a calmer face.
That's right.
Right. Avoiding the weird. Okay, in this sense, maybe what it implies to me is that you're doing this mentally. Yeah. Which is good. Again, a worrying activity and avoiding it, yes, can be done mentally rather than physically, of course. So you're saying, I'm not going to stress about this.
And I would say the one lacking component here is that I don't get the sense that there is or was an actual worrying activity.
Yeah.
That this person is just in a sublime mental state.
Yeah. So the thing lacking here to give clarity it has to include something that shows the worrying activity of some level, rather than this could just be interpreted as calmness and
Right.
enjoying a calm place.
Right.
Wow.
That ball and chain wrapped around the waist.
Wow.
Toss it to the wind if you can.
And the emoji head on that emoji body.
Wow. That's great. That is fun.
Yeah, that's fun too.
That is fun. Well done. Oh, interesting. So the octopus is dreaming of gathering that apple.
Wow. Again, it's interesting that now two people have gone outside of the realm.
Yeah,
So
to be terrestrial.
this octopus is actually privy to knowledge outside of its world
Oh my,
to be able to dream about it.
dreaming the unimaginable, the
Yeah.
unknowable,
Very good.
unless the octopus is a captive octopus in a lab and spots on a television set an ad for apples from an apple tree. We can only guess. Dreams beyond reckoning.
Oh, wow.
Oh, there's the worry.
Clear worry. The
Yes.
posture is so clear.
Oh, you tackled a couple at
Yeah,
once.
okay.
Thought about two of them at once.
You'll see in my example that I also used your method that you're using there to show graphically increase, decreases, and staying stable. Yeah.
Oh, an ostrich avoiding avoiding worry. Stick that head in the earth.
This is a very interesting one because again, this is one of the things that I had to think about. How many people on the planet would be familiar with the concept of an ostrich sticking its head in the ground?
Yeah.
And not just me, but the group that's going through this process of trying to decide which emojis will work or not has to say, is there enough people on the planet that would be using this app that would
Right.
recognize the ostrich with his head in the sand or
Right.
not?
And that'd be, if we had time, that'd be a fun pop poll.
Yeah.
Oh, here we go. Get away from that worry.
Again, as you saw from my emoji art, I did feel that I had to go beyond just a face. I used that whistling mouth as well for another emoji that I did that was much simpler, not with that complexity of avoiding a worrying activity, which has two levels.
And we'll see that shortly.
Yeah.
Oh, this is great.
Well, Barrett, I just wanted to mention we have about 5 minutes left, so
Okay.
if you want
Okay.
to share your representations of that.
Yeah,
Very good.
let's do it.
Will do. And
All right.
another.
Nice.
Oh, interesting.
Oh, yes. Octopus dreams.
Yes, I see the arms of the octopus. Suggestive arms, possibly?
Something that Barrett and I have wondered about, whether an octopus would dream with itself in it or only dream with it seeing its tentacles, since it would never, well, it could dream about another octopus it saw.
Right. Oh, this is lovely. You've
Oh,
tackled both the number
very,
2 and number 3.
Very beautiful for the worry.
Yeah.
Just the drawing is wonderful. Yeah.
Yeah. And maybe the avoidance is the turning away from the source of harm or
And the number 3 also is a very good starting sketch that would be obvious that they could move along with that too. Oh,
wow. That's gorgeous.
okay. So that's something done in 20 minutes. Hard for us to even imagine.
Oh my goodness, beautiful work. Could you put in the chat who you are?
Woof.
Now, the octopus has, oh, interesting. Yeah, well, I won't spend time with this except to say that's gorgeous.
Yeah. Mm.
Emoji.
We've already commented about this in various manners. Yeah.
Good.
Ah, yes.
I see it.
Again, the split between the two,
Yes.
the two sides, very
And
good.
look, you've got the potassium, nitrogen, phosphorus
Yeah.
right there laid out symbolically.
Oh,
Oh boy.
That's another
It's
nice-
food.
yeah.
Yeah, actually showing the sleep action. And
What do you know from other animals? They're wondering about.
oh,
yes.
interesting. Yeah.
Well, there's a lot of people wondering about sleep.
I'm still hungry. Sure enough. Nope. Nope. Oh, interesting.
Yes.
Getting that crab out of the jar. Beautifully done. While asleep. The thought bubble. Very nice. Oh, another gorgeous work with an octopus.
All right. Water.
Dreaming of another octopus. of nothing the world.
Clearly
can
this
do.
person was inspired by the talk today because they quickly made some inspirational art there.
Yeah.
Oh,
Uh-oh.
that's nice.
Yeah.
Avoiding, yeah.
Oh my goodness. Wow.
Dream.
All of our dreams right there, encapsulating that image. Oh, nice. A more surreal suggestion.
Oh, yes.
Yep.
Oh.
Avoid that. Avoid that.
Yep.
Worry. Dream among the birds,
Of
being
birds again.
floating up in the clouds.
There is some sense that they want to fly.
Another. Very good.
Oh, right.
I like that.
This one is also very well done. Nutrient density. So they're defining how their drawing is with a nutrient density bar. Yes.
Oh, interesting.
Oh, yeah.
Oh, love it.
Yeah. Also beautiful.
Stylistically
A
beautiful.
brain.
And
we
this
gotta
is the
go.
last one. Okay, so now we're going to share with you what our visions were.
No more spectacular than yours, that's for sure.
That's right. We loved your concept pieces. Now skip ahead and okay, visualization challenge number 1. This was how my mother interpreted it, at least with the input from John Laskew, Niels Rattenborg, and me, with, we have no idea. So why don't we show something like nothing on the first third, and then food, or what they imagine would be fitness increasing that they might think about a lot, attaining food, as several of you showed, and then maybe something surreal, abstract, as others of you suggested.
Yeah, beautiful.
Okay, second visualization challenge, the emoji and how Damon tackled it.
And as you can see, I tackled it in a way that many of you did as well. So including that individual running away from something. I thought this wasn't something that popped in my head immediately. I had to think about this for a while, and it feels effective. The only thing that I think I did more effectively than a few that only had 20 minutes and I had plenty of time was really showing a worrying activity that I think many could imagine, that they have that microphone in their face and they're expected, and they're in the spotlight. They're expected to do and say something, and they want to get away from that.
And the last challenge, which many of you took on, which was brave, I thought.
Not only brave, but you guys, many of you did the thing that I should have thought of immediately by separating into 2 parts. That world that we existed in 100 years ago to that world we exist in now with higher CO2. So that is a very effective tool that I didn't use. The tool that I did use strongly here was showing graphically the going down of nutrients per leaf, the increase in the plant biomass, and the soil nutrients not changing, without having to do it in any strong graphical way.
So you did a noble job, your efforts. I applaud you all, and thank you so much for participating. I'd like to add to the discussion that none of the works, including the last 3 we showed, are perfect. We each tackle visualization challenges in unique ways, artistically, because we've got several factors to achieve success, graphical excellence. One has to be accuracy, first and foremost. Second has to be clarity. And third, that efficacy comes out through aesthetics in an ideal sense. But the subjective nature of what makes something aesthetically appealing varies, of course. So always think about your audience. What does your audience require? Who are the people that you're trying to reach when you're trying to establish your visualization goals? And we're 2 minutes over time. If anybody has last-minute comments or questions, I can pull up the Q&A. Otherwise, Joanna and Daria, I assume we have to wrap up. Thank you, everyone. I've loved being involved, thrilled and honored that Damon was able to join in for workshop number 4.
Real pleasure to be able to share in that process with all of you.
Well, thank you so much to our speakers and for all of you who are participants. We will post the link if you want to still upload your work, if you weren't able to, and any resources from today's session, we'll make a list and make that available. All right. So thank you everybody for participating, and we hope to see you next time for a workshop on making logos.
Yay! Thank you again, Joanna and Daria. This has been such a blast. And I love that so many of you have joined from so many different places around the world.
Hey, there's your mom.
Yay! There's my ma. You got to see so many of your works, ma. Like, I can show the original for those who are still hanging around of the Octopus Dream. And you see how something that I couldn't show on PowerPoint was the gold in the surreal component, an exquisite part that sadly is lost in digital representation.
Yeah.
So this is a case where the tangible component differs from what the masses will receive
That's
in the
true.
ultimate book form and format.